Hello students. So today through this video I shall be talking about ISC specimen question paper 2024, which was released by ISC official site, and I already discussed about section A. See that video, which is mainly focusing on section A, the questions and their solution. Now in this video we will be talking about section B, which mainly consists of 7 questions of 2 marks each. So let's begin. But before that, don't forget to subscribe my channel. And if you like this video, after seeing that video, if you feel that it will be helpful for you, then you please share it. So let's begin for section B, as you already finished section A in my previous video. So in section B, directly we are moving to section B. Okay, it is here. So section B, total 14 marks. Actually, each question carry 2 marks. So in this part, they will give you 7 questions. And obviously, there is no internal choice according to new board pattern, according to new question pattern. So first question, a couple does not wish to have more children. Suggest and briefly explain any 2 methods of family planning that are highly effective and have low chances of failure for the couple to consider. So that means they are talking about the family planning program and how we can chalk the population. So there is the contraceptive methods. So I wrote 2 methods here through which the family planning is, we can apply these methods to control the population, and this way we can maintain the family planning program. So I wrote 2 methods. One method is barrier method. So barrier methods are mechanical methods by which sperm and ovum are prevented to come in physical contact. The common barrier methods are condom, fam shield, diaphragm, cervical cap, and bolt cap. So this is one important method by which we can check the unwanted pregnancy. Another is the surgical methods, and that is the sterilization provides a permanent and sure method of birth control. It is known as vasectomy in case of men, because in this case, in men, we have to remove the vas deferens of sperm duct. So surgical way, we can cut the vas deferens. And another case where fallopian tube is mainly removed in case of women. So this method is tubectomy. So 2 main methods in surgical methods are vasectomy and tubectomy. And these 2 methods, barrier methods and surgical methods, these are highly effective and have low chances of failure. Okay. Next question. In a population of 5,000 individuals, 1,800 do not have freckles on their faces, small f, while the remaining individuals have freckles, capital F. That means those who have freckles, that is capital F, and where the freckles are not there, we are considering it as small f. Assuming that the population is in Hardy-Weinberg equilibrium for the presence of freckles, calculate the expected frequencies for the following genotypes. So it is mainly about the population genetics. It is given from evolution topic. So now they are asking the frequency for the FF genotype and small f genotype. So how we can estimate or we can determine the frequency of FF genotype? In this case, there is the formula, number of F alleles divided by total number of individuals. So number of F alleles here, as it is given 5,000 total number of individuals in a population, and small f value is here 1,800. 800 where the freckles are absent, and so the rest, that means 3200, that means you need to subtract 5000 minus 1800 and you will get 3200, where this 3200, that means the number of individuals who has the freckles, capital F. So here number of F alleles I have written thus 3200, and now we need to divide them by total number of individual 5000, and we will get 0.64. So this is the frequency of FF genotype. Now to see the frequency of small f genotype, same way we need to follow the formula, but in this case we need to take number of small f alleles. Previously I took number of capital F alleles. Now to determine small f genotypic frequency, we are taking number of small f alleles, and here it is given 1800, and then divide it by total number of individuals, which is 5000 already given, and this we will get 1800 by 5000, that means 0.36. So that is the frequency of capital F genotype, it is 0.64, and the frequency of small f genotype is 0.36. This is the answer for this question. Next, shown below is the karyotype of an individual. So in this case, if you see 1, 2, 3, that means 23 pairs of chromosome is showing, but there is no problem up to 22 pair, but in the last pair, in 23 pair, we can see it is not in diploid form, it is in the haploid form. That means one chromosome is deleted from the genome. Okay, so now by seeing this diagram, the question which they are asking, we need to focus on it. State one characteristic reproductive feature and one physical attribute of such an individual. So in this case, in the reproductive feature, when we will get such kind of chromosomal abnormalities, that means where the X chromosome or one sex chromosome is absent, it is indicating the monosomy of X or sex chromosome, mainly here X chromosome. So what kind of reproductive feature we can characterize there? So reproductive feature in this case, we will see underdeveloped breasts and small uterus. And they also asked to write one physical attribute. So one physical attribute that we will get for this chromosomal pattern is that it will be sterile female with poorly developed ovaries. So this is reproductive feature, and this is physical attribute or physical appearance, physical feature. Next, related to this diagram, they asked what is the category of such disorders called. So it is actually called Turner syndrome, where one X chromosome is absent. So we can say it 2n-1, that means monosomy of sex chromosome or X chromosome. Now, which abnormality during the cell division causes such disorders? That means what is the reason behind this kind of appearance, this kind of situation, why it appeared? So it appeared due to the non-disjunctioning of X chromosomes during meiosis division. Move to next question. Study the diagram of a human ovum given below and answer the questions that follow. So identify, so this is the structure of ovum, and they have labeled it as 1, 3, 4, 1, 2 and 1 and 5, but they didn't ask anything about the labeling. So let's focus on the question what they asked. Identify and name the part of the ovum that prevents its fertilization by multiple sperm. So the answer for this question is zona pellucida. Zona pellucida. Now, what is the mode of action of the part identified in A? That means in question A, what is the mode of action of this part? So it is written, as the first sperm comes in contact with the zona pellucida layer of the ovum, it induces changes in the egg membrane and egg cytoplasm. Firstly, it blocks the entry of additional sperms, and secondly, the egg cytoplasm extends the head of sperm and forms fertilization core and finally engulf the sperm. So this way the zona pellucida is mainly preventing the entry of extra sperm after the entry of one sperm inside the ovum. This is the mechanism of action of this zona pellucida layer. Move to next question. The diagram given below represents a specific stage of human embryonic development. Study it carefully and answer the questions that follow. So this diagram, by seeing this diagram, which stage of the development of the human embryo is mainly representing here. So the name of this stage is blastocyst or blastoderm. Identify the part labeled A. You can see level A where it is labeled, actually the part A where it is actually labeled. So what is the name of this part A? So it is actually trophoectoderm. It is trophoectoderm. What happens to the inner cell mass after implantation? That means this inner cell mass, which is they have shown here in this diagram, what actually happened after its implantation? Implantation means fixing of the embryo to the wall of the fertilized egg or the zygote with the wall of the uterus, wall of the uterus. So in this case, that inner cell mass which mainly formed in the blastocyst, what will happen with this inner cell mass? How it is transforming into a new structure? So let's see. With the appearance of blastocyst, morula changes into a hollow blastocyst by 5th day after fertilization. At the end of 5th day of fertilization, blastocyst enlarges and pushes the inner cell mass to one side. It forms a knob in blastocyst, which is called embryonal knob, that finally gives rise to embryo. So this is the fate of this inner cell mass after implantation. Next question in section B. A group of researchers in a pharmaceutical company aims to produce a specific chemical which can treat viral infections. Which specific group of chemicals should they isolate, and how would it help in treatment of viral infection? So the specific group of chemicals that they should isolate is interferons. How would interferons then help in treatment of viral infection? So when these interferons released by the infected cells, this interferon makes the surrounding cells resistant to secondary viral infection by inhibiting multiplication of viral particles, by inhibiting viral replication. So this way they will inhibit or they will stop the entry of new virus, okay, the entry of the secondary viral infection. Next question. Explain any two types of evidence for biological evolution. So one of the evidence is evidence from paleontology. So paleontology is the study of fossils of animals and plants that lived in past. Fossil records have helped in building the broad historical sequence of biological evolution. Another evolutionary evidence is that evidence from comparative anatomy and morphology. So comparative study of morphology and anatomy of various organs and organ systems reveals that both similarities as well as differences exist in the structure of body organs due to the similarities and differences in their functions. So these are the two evidences that I wrote, one for paleontological, one evidence for biological evolution is paleontological evolution, and another is comparative anatomical and morphological evidence. Move to the next question of section B. The figure shown below is a representative image of an antibody. Study this diagram carefully and answer the questions that follow. So this is the structure of antibody. So a person is infected by a pathogen. Identify and name the part of the antibody that the pathogen binds to. So first is identify that antibody which will be responding when the pathogen enter or when the pathogen infect any person. So it will be IgG antibody. IgG antibody. And which specific part of this antibody mainly binds with the pathogen? It will be known as Fab. Fab, the full form is antigen binding filament. Next, a few years later, this person is infected by a different variant of the same pathogen. Would the existing antibodies be able to recognize and bind to it now? Justify your answer by giving one reason. So the answer is yes. IgG molecules stimulate alternative pathway of complement system and act on opsonins to stimulate phagocytic activity. Antibodies called opsonins get attached to the surface of the antigens. This enhances their recognition and digestion by the phagocytes. So this is about section B, where I covered 7 questions, where I solved 7 questions of section B. So hope this video will help you for your 2024 board exam. These are a few questions. These are some demo questions. By seeing this question, you can assume what type of question they will give you in board exam. So please don't forget to subscribe and share this video. Thank you for watching this video.